So good evening. We are Alejandra, Fernández, and Haritz Plazaola. We are PhD students for the University of Barcelona, the Department of Genetics. And first of all, we want to thank the organizer for giving us the opportunity to present today our work. And the talk that we are going to explain you is called Wiring Specificity, How the Neurons Are Connected. Maybe you are wondering what does wiring specificity means. As you know, the neurons in our brain are connected one to each other in a specific manner, creating a huge and sophisticated network. To make easier the understanding, is better if you compare with a computer and its circuits. These circuits need to be properly assembled for the correct functioning in both cases, in the case of our brain and in the case of the computer. A cable or a neuron has a specific partner to connect. And if, for example, you connect the mouse in the computer in the place of the headphones, it doesn't work.
The mentioned circuits must be properly assembled, and if some of those connections fail or the neurons die, we'll have neurological diseases with catastrophic consequences. This happens because the system cannot process the information properly, and some of the neurological diseases are Huntington disease, schizophrenia, Parkinson, autism, or Alzheimer. And right now, no one of them have a cure. In the way to find that cure, nowadays we can already obtain neurons derived from stem cells, but in the future we will be able to replace these neurons and reconstruct the lost connection specificity between them. So this will be a future application of our work.
Behind the wiring specificity, there are the genes. The genes are going to play a key role in this process because each neuron has a set of genes that make them different. Our objective is to unravel the genes that they are going to be essential for a neuron to find its partner. So, knowing that, we will be able to reproduce and reconstruct the pattern of connectivity that are lost in our brain. For this purpose, we use Drosophila melanogaster as a model, and we have a very important reason to use this animal as a model, because it's very cheap and also is very fast to do the experiments, and also we have a ton of powerful tools for genetically modify the flies, so we can mutate a specific gene in a specific tissue to study the function of the gene. And also another very important reason is that we share a huge percentage of genes with them. And we have a very similar neuronal structure, so this means that maybe a gene that is important in the fly also is important in humans.
And in the fly specifically, we work in the compound eye. As a curiosity, the fly compound eye has more or less 750 small eyes. So as they have two compound eyes, they have more than 1,500 of them. So you can imagine that they see pretty well. Each small eye has special neurons that are responsible for transmit the visual stimulus from the eye to the brain. To situate, you can see in the right an image of the head of the fly, and inside it, everything that is colored is the brain of the fly. In the corners, there are the compound eyes, both of them, and there will be located the photoreceptors, where the photoreceptors are those specialized neurons that are in each small eye to send the light information to the optic lobe that are in green. So the approach that we are taking to address wiring specificity is to study closely related neurons that share developmental origin, so in our case photoreceptors, and has similar functions, but they have different connectivity. Our hypothesis is that most of the genes that are differentially active between those closely related neurons will be used to generate this different pattern of connectivity.
In this slide, you can see in the left part the two photoreceptors that we are going to study. The one that is red is the photoreceptor that is called R7, and the one that is in blue is the photoreceptor that is called R8. And as you can see in this scheme, they have different connectivity because they connect in different layers in the brain. The picture that is on the right is a real situation. It's a section of the brain of the fly, and you can see, as you can see again, the photoreceptors are 8 and the photoreceptors are 7. Thanks to bioinformatic tools, we have the information about the genes that are differentially active if we compare these two types of neurons. So, for example, in the case of the R8 photoreceptors, we know that there are more than 200 genes that are specifically active in these cells, but they are not active in the other photoreceptor. So, this means that these genes could play a key role in the connectivity.
As we said before, the Drosophila is a powerful model because we can remove one gene from a specific gene without affecting the surrounding cells. And this we can do it in vivo. And also, we can label the neuron of interest in the color that we want. For example, here you can see those in green. As an example of removing a gene, here in the image of the right, you can see what happens when we remove a specific gene from the neuron of interest. So those cells, those neurons mistarget to another layer. So in few words, instead of going where they should be, they stop earlier. You can compare it with the image in the left, and you can see how they stop earlier. Seeing that, we can conclude that the gene that we removed is important for the specificity of these connections.
As a summary, as all of you already know, the connection specificity is very important to process properly the information, and we use to study this the fly visual system as a model.
And the photoreceptors are neurons that transmit visual information to the brain, and our main goal is to discover genes that control connection specificity. And this could have future applications like neuronal replacement. And thank you for your attention.